Hello. Yeah. So sorry, Dr. Sridhar, for the delay. We just want to introduce our participants as well. That this is a special online lecture series initiated by Gujarat State Biotechnology Mission, Government of Gujarat, along with different partners like NIPER, IIT Gandhinagar, Indian Institute of Public Health Gandhinagar, in the name of City Academic Circle. Okay. So this is a City Academic Circle which is organizing such online events as well as physical meets with the researchers and academicians every month. So this month we initiated this lecture online talk. So everyone, today we have with us Dr. Sridhar Desikan. So Dr. Sridhar is a pharmaceutical industry executive with over 24 years of diverse experience in innovator and generics pharma industries with global experience in both USA and India. Currently, he is a global technical head formulation R&D at Piramal Pharma Solutions, USA. He is leading the formulation R&D organization for Piramal Pharma Solutions with CDMO and internal product development responsibilities with teams across 4 R&D centers in the USA, UK, and India. Prior to Piramal, he was Executive Vice President Formulation R&D at Dr. Reddy's Laboratory. He led R&D teams responsible for generic products for Dr. Reddy's Laboratories across diverse dosage forms, oral solids, injectables, and inhalation products with specific focus on complex generics. He was a part of leadership team of Bristol Myers Squibb inclusive USA to set up an R&D center in Bangalore, India, in collaboration with Syngene Biocon. Okay, he has several patents and research publications. There is a long list in the introduction of Dr. Sridhar, but I would cut short it. And now it is a time wherein we can hear Dr. Sridhar. So Dr. Sridhar, now platform is yours. I would request you to take you ahead. Thank you.
Thank you so much for the introduction. And just want to share my screen. Are you able to see my screen?
Yes.
Yeah, so thank you very much for the invitation today to speak. And I'm really thrilled to see such an academic circle, City Academic Circle, being organized by interested scientists across Ahmedabad, across Gujarat. And thank you very much to Ravi Shah, Dr. Ravi Shah, for inviting me to give this talk today. And so today I'll be talking about prodrugs in drug development to address bioavailability. Started about maybe 15 years back when I was with Bristol Myers Squibb. At the time, I was part of the team that was responsible for enhancing the oral bioavailability of poorly soluble compounds. And as many of you can appreciate, many drugs that enter into development have very poor solubility, very poor permeability, or both. And this poses significant delivery challenges. And my team at BMS was responsible for addressing the dissolution rate limited and solubility limited challenges. So we came across technologies such as hot melt extrusion, spray dried amorphous dispersion, and nano suspensions to overcome some of these challenges. However, I always used to wonder, wouldn't it be great if we have a chemical modification to address some of the delivery challenges, because the challenges are not associated only with the solubility alone. And hence my interest in prodrugs started. Later, as part of the leadership team at Biocon BMS R&D Center, the R&D center that we set up in Bangalore, we had the opportunity to create a multidisciplinary team of medicinal chemists, synthetic chemists, DMPK scientists, pharmaceutic scientists, and also formulation experts together as a team, an expert team to address systematically the delivery challenges with respect to new molecule entities that have very poor bioavailability. So in my talk today, I'll be talking about some of well-published literature in the topic of prodrugs. And for those of you who are interested to learn more about it, I would highly recommend an excellent book by Valentino Stella and edited by Stella and team. And my mentor, Mike Hageman, who is currently a professor at University of Kansas, is one of the editors of this book. I like it so much that I use it as a prop for my computer at home. So, so with that as an introduction, I want to just quickly get into the topics for the discussion today. And as I indicated earlier, I'm going to be talking about as a personal capacity. I'm not representing Piramal Pharma Solutions in this talk, and all the material that is published here are in public domain. And I would appreciate if this content is not disseminated widely without my consent or permission. So when you talk about drug delivery challenges, as I had indicated, drugs are discovered through library synthesis, combinatorial chemistry, structure-activity relationships, and other techniques. And initially, in the drug discovery paradigm, we have close to maybe 100,000, even 20,000 molecules that are screened for specific activity towards a molecular target. And then the compounds go through various screening assays, both for purity, solubility, stability, permeability, metabolic lability, and these then get winnowed down to very few candidates that are taken up for further for development. So when you speak about drug delivery challenges, there are multiple drug delivery challenges at each stage of development. And what is considered as drug delivery for the primary activity assays is essentially having a formulation that can be used for screening wide array of compounds with different physicochemical properties to be able to get accurate, reliable activity assays. In this case, it could be something as simple as formulating as a DMSO, DMSO solvent as a solution with very high solubility to be able to have a formulation that can be easily dosed into these multi-well format to get the primary activity assays. And some of them could be enzymatic assays for which the formulation composition has to be compatible. And as you go further down in the screening assays, you get into the secondary in vitro assays. These could be specific cell-based assays to understand the mechanism, and some of them could be in vitro disease models and bioassays that are used to understand the efficacy of the molecule that we are developing. And then, of course, as we progress further down in development, we go into the in vivo activity assays in the preclinical models. And for these, the pre-formulation efforts and having an appropriate formulation to deliver, to get the accurate evaluation of the compounds become very important. After we go through all these things, we finally end up with one or two molecules that are taken further for development. So if you look through the literature and overall drug development, and those of us who have been in the industry for quite some time, we appreciate how difficult it is to get a new molecular entity in the market. And a review by Waring et al. That was published in 2015 surveyed 605 compounds from 4 or 5 major pharmaceutical companies, and then evaluated them as a reason for them not progressing or getting into the market. And if you take a look at the overall number of compounds, all through at different stages of development, many of them get winnowed or get terminated because of toxicological reasons, and of course the business or reasons also play a significant role. And the reason for the overall reason for attrition of the compound due to pharmacokinetic and poor bioavailability is not insignificant. It is significant. And this is even further highlighted for the compounds that progress to phase 1. The compounds that enter phase 1 but do not progress forward, a subset of these molecules out of 605, about 160 compounds that we have, And out of these, about 40 of them get terminated because of poor clinical safety. And about 25 of them, if you think about it, close to about 1/6 of the total number of compounds get terminated because of poor pharmacokinetic and poor bioavailability. So this high attrition in this phase one, is something that can we address that so that we do not lose the compounds that are highly promising. Otherwise, is there any way we can make them druggable and marketable? And so that is the challenge that we are trying to address through interventions such as prodrugs. And so in this case, the highly low and highly variable bioavailability is one of the main reasons for stopping for further development. So if you look at the overall, barriers to oral drug delivery for systemic exposure, so I just want to kind of, through this cartoon, I just want to highlight the various rate processes that are involved in drug delivery. When you take a drug orally, whether it is a tablet or a capsule, that is a formulated drug product, it undergoes the disintegration first, and then the dispersed drug product. These could be, the granules that contain the drug substance, then dispersed into the in the gut, and then it undergoes the dissolution. And there is a rate process of both the dissolution and precipitation, depending on the solubility properties of the drug. And once the drug is in soluble format, it is then taken up. It is permeable and it is absorbed. And in this absorbed drug, that is, that reaches a target organ for it to be effective. So for an oral drug, it passes through the gut, goes through the, and the first, barrier that we have is a gut wall. It has many enzymes that are present that essentially, have metabolized the drug. We call it as a gut wall metabolism. And then the partially or the remaining fraction of the drug gets into the portal vein and taken up by the liver, where it is further, metabolized, and the fraction that is not metabolized in the liver and then passes into the systemic circulation, and that is bioavailable, and then it's a free drug. Then there is not protein bound that reaches a target for activity. So if you look at the overall, oral bioavailability, it is a function of a fraction that is absorbed and the fraction that is extracted through the first pass metabolism. And the overall efficacy is a combination of the intrinsic potency of the molecule and the oral bioavailability. And so this, in essence, is as you can see, there are multiple challenges to oral drug delivery. And in the case of parenteral drugs, and this first pass metabolism is overcome, and the drug is administered directly into the bloodstream. And however, in the case of the parenteral drugs, injectable drugs, solubility challenges are important. To be able to administer the drugs in a reasonable volume, you need to have adequate solubility, and the stability of the molecule in the formulation become critical barriers for the drug to be delivered intravenously. So if you look through the formulation challenge strategies to overcome dissolution rate limited or solubility limited bioavailability, there are two major classifications or two classes of interventions that development scientists undertake. The first is solid state modification, and this is addressed either through particle size reduction, whether it is micronizing the material or nanosizing. This essentially addresses the dissolution rate limited bioavailability to increase the dissolution rate of the drug to make it soluble. And then we have non-crystalline interventions such as spray-dried amorphous dispersions and also hot melt extruded formulations, wherein the crystalline lattice is destroyed, and the initial step of the lattice energy is circumvented, and to make it available, in terms of enhancing the solubility of the drug. And then we also have the solubilized formulations where the lipophilic molecules are formulated along with other additional lipids and can be dosed as solubilized formulations. And then, of course, we have other strategies such as salt formation in different polymers, hydrates, and co-crystals in solid complexes. And these are the various techniques that are used for overcoming solubility limitations from the solid state perspective. From the solutions side, we have approaches such as controlling the pH and addition of the surfactants, use of the co-solvents and complexing agents such as cyclodextrins. And I mentioned about the lipidic formulations and combination of all of these, for example, things like liquid-filled oral capsules are approaches to address the solubility limitations through solvent modification. And what I am going to talk about today is chemical modification to address these. And these are the prodrugs that addresses both the solid state and also eliminates the need for the solvent-based modifications. And then, of course, the barrier to delivery is also through permeability limited bioavailability. If the compound is poorly permeable, even though if the compound is highly soluble, if it is limited by permeability, then it is not going to be bioavailable. To address that, there are, if you talk about permeability, there are two major, aspects of permeability. One is passive permeability, that is passive transport limited. And in this case, there are approaches where we can have a high supersaturation, so to drive the flux of the drug through the membrane to make it more bioavailable. And there are strategies to use the permeation enhancers, and this is still of questionable utility, as there is a likelihood of high variability in use of permeability enhancers in the formulation. And there are drugs that are efflux pump limited, and many of you are familiar with efflux pumps such as polyglycoprotein PGP efflux that essentially take the xenobiotics out of the cells and efflux out into efflux out of the cells. And so in these cases, there are approaches to saturate the efflux pump. And again, in this case, this is of questionable value. And there are strategies, there are drugs in the market where we have combination products of the efflux pump inhibitor along with the drug of choice to be able to deliver your desirable drug. Here again, the prodrugs to overcome the permeability challenges are a good approach to address permeability-related bioavailability. So what are prodrugs? So prodrugs can be considered as bioreversible derivatives of drug molecules and that undergo enzymatic or chemical transformation in vivo to release the active parent drug. So essentially, if you talk about a drug, drug by itself is not permeable or has the barrier. This barrier could be many different types. It can be solubility, dissolution rate, or it could be permeability. And these barriers are overcome by a prodrug, which is essentially a chemically linked a drug, a covalently linked molecule. And then that gets go through the barrier. And in the body, in vivo, it undergoes enzymatic or chemical transformation to release the drug, and that is then made, that is then bioavailable. And so this, in essence, is a crux of the drug delivery using the prodrug. So when you talk about the prodrugs, why prodrugs? I mentioned about the prodrugs to overcome the aqueous solubility, poor permeability, first pass metabolism, and also poor targetability. There are prodrugs that are designed to target specifically in a specific organs. I'll be talking about that later in my presentation. And what I am showing here in this plot is a modified biopharmaceutics classification system, and that many of you are familiar with. In this case, what I am sharing here is a developability classification system. Typically, in the biopharmaceutics classification system, we have solubility and permeability, and then you classify the drugs based on class 1, class 2, class 3, and class 4 as highly soluble, highly permeable, poorly soluble, highly permeable, and highly soluble, poorly permeable, and poorly soluble, poorly permeable as BCS class 4. However, in the drug disposition and a developability classification system, it is important to look at it from the perspective of the dose number, essentially a dose solubility ratio. So the reason why the DCS classification system was developed is that the bioavailability is not just a function of the solubility alone. It is also a function of the dose. Compounds that have a very poor solubility and can still be bioavailable if the dose is very low. And likewise, on the flip side, even a compound that has a good solubility, but if the dose is very high, can be very poorly bioavailable. So what is shown here is also within the BCS classification system, in the DCS classification system, class 2 is further subdivided into two, into a dissolution rate limited bioavailability, that is a class 2A, and solubility limited bioavailability, that is class 2B. And SLAD is a line that is a solubility limited absorbable dose that is calculated as a small intestinal solubility and times the volume and the permeability parameter, typically an absorption number, to give you the line of the solubility limited absorbable dose number. So in this case, what I've shown here is a few of the doses for a given drug with a given permeability. At the low dose, it behaves as a class 1 molecule, and beyond a certain dose, it gets into the dissolution rate limited bioavailability. In this case, for this compound, even at the highest dose, it is on the dissolution rate limited side, and hence the bioavailability may not be a significant challenge. And for the prodrugs case, our objective is to transition compounds that are in the solubility limited bioavailability in the class 2B compounds and make them soluble enough to get them into the class 1. And likewise, there are compounds that are very poorly permeable and poorly soluble, transition them from class 4 into class 1 so the delivery challenges can be overcome. So that is the objective of the prodrugs that we are working on. So prodrugs by the numbers. If you really do a survey of the recently approved prodrugs over the past 10 years, about 12 to 15% of all the FDA approved small molecules are prodrugs. And this is a significant number. So in terms of the number, we have about 30 prodrugs approved over the past 10 years. And it is really fascinating to see the diverse range of prodrugs that have been marketed. And by a reasonable estimate, we can say that about 10% of all the marketed prodrugs all the drugs or the small molecule drugs can be considered as prodrugs. Here, I'm not even referring to macromolecular prodrugs, antibody drug conjugates. Some of the prodrug experts consider the antibody drug conjugates that are very common in recent approvals. They are nothing but prodrugs because they are conjugated through chemically, covalently to release a drug. And even without considering those, we have significant number of the prodrugs that are marketed. In terms of the patent literature, the reference here is slightly older. There is a steady increase in the number of patents of the prodrugs. Increasingly, the trend for the new molecule research groups is to publish the initial patent to include the prodrugs itself, to have the IP coverage, to include the salts and the prodrugs of the parent molecule in the first composition matter patent itself. So the common types of prodrugs, I just want to do a quick check. It's about half an hour into the presentation. I will quickly go through the rest of the presentation. There are multiple types of prodrugs. For the prodrug to be made, we need to have a prodrug handle. The prodrug handle can be a carboxylic acid, thionyl group, hydroxyl group, or a phosphate group, or the amine group. And these are the handles from which the multiple different diverse prodrugs can be synthesized. So just I want to quickly go through some older examples and the new examples of prodrugs. The first example that I can share with you is prodrugs for improved permeability. What I am sharing here is the privampicillin. Is an beta-lactam antibiotic. It is a pivalloyl methyl ester of ampicillin. So essentially, we have the pivalloyl methyl ester and that is present in the drug, and it undergoes the conversion through esterases to release the drug ampicillin, and also releases the other formaldehyde and pivallic acid. So if you look at the serum concentration of ampicillin by itself versus the pivampicillin, you can see the oral bioavailability of the ampicillin itself is about 30% or so, and it is improved to 87 to 94% for pivampicillin. And this is a very classic example of permeability enhancing prodrug that has been marketed. Likewise, elanapril is the monoethyl ester of enalaprilat. And here in this case, we have the monoethyl ester that is bioconverted through carboxyl esterases to release the parent drug enalaprilat. And here again, the oral bioavailability has been significantly enhanced with the prodrug, with about 75% absorbed because of the increase in the permeability. And other interesting case here, I want to talk about this in a little bit more detail, is the nucleoside inhibitors. Tenofovir is one of the examples. For the nucleoside analogs, the nucleoside analogs here to be active for, in the case of HIV inhibition, they need to undergo, phosphorylation in the cells for it to be recognized by the viral RNA for it to be effective as a drug. The first step of the process is what is called a monophosphorylation, is a rate-limiting step, and the phosphorylation happens in the in the cytosol in the cell, going from with the help of kinases to go from a monophosphate to a diphosphate to the triphosphate. Since the monophosphate is a rate-limiting step, the prodrugs have been synthesized to have it as to deliver as a monophosphate as a drug. However, because of the phosphonic acid groups, the polarity of the phosphonic acid groups, they are not permeable, and hence the drugs have been synthesized to make them as a permeable version of the monophosphate. In this case, it is the isopropyloxycarbonyloxyl methyl group. It's a bis pivalloyl group here, isopropyloxyl group, and of the tenofovir that I'm showing here. And this increases the permeability significantly. So the oral bioavailability of the tenofovir, when dosed as the disoproxyl prodrug, is about 39% in the fed state. And without this, the delivery of this phosphate group itself would not have been possible. So this is a very, very classic case of using prodrugs for enhanced permeability. And likewise, there is when we are talking about the prodrug here, I want to share with you a very recent advancement in the permeability limited or permeability enhancing prodrugs. Here, this is what we call as I guess the ProTide technology. This is again a tenofovir, but in this case, the pro moiety is alafeminate prodrug, and Genvoya and other combination drugs using the tenofovir used in the antiviral and HIV treatment uses this prodrug. Here it is an aryloxy and phosphoramidate pronucleotide, and it is an isopropyl ester group here that I am showing you here. And in this case, the activation happens intracellularly. In the case of the tenofovir disoproxyl that I spoke about just now, the activation happens in the plasma where it gets converted. And in the case of the alafeminide prodrug, because of the presence of the isopropyl ester group, it gets permeated intracellularly. And inside the cell, this the group gets cleaved, releasing the monophosphate group, which then subsequently undergoes the triphosphorylation for it to be effective. This ProTide technology using the aryloxyl phosphoramidate prodrugs has been tried for quite a few prodrugs recently. There are at least 4 or 5 prodrugs that I'm aware of and that uses this ProTide technology. And in this case, because of this intracellular targeting, we are able to achieve a very high permeability cellularly. And so a 10 milligram of a tenofovir alafeminide is equivalent to 300 milligram of disoproxil prodrug. And this is a very interesting new example of cellular targeted prodrugs. And when do permeability enhancing prodrugs work? So when we have intestinal permeability of the parent as a major barrier to bioavailability, those are the cases where the permeability enhancing prodrugs are important. And for this to be effective, the parent drug needs to have low to moderate clearance. Reason why I'm saying this is because if the compound is very highly cleared, then even if you have a prodrug deliver the parent drug, it is rapidly eliminated, leading to drug not being available, and hence, in this case, the permeability enhancing prodrugs might not be effective. And in the cases where the liver extraction ratio is higher than 0.5, the permeability enhancing prodrugs might not be effective. And so, for the drug to be, reaching the, for systemic circulation, the first pass metabolism or the, liver extraction has to be low enough for it to be viable. And the parent drug itself, should not be solubility limited. And there have been instances where we have seen in the preclinical models the drug that is delivered as a permeability enhancing prodrugs upon conversion to parent in the in the systemic circulation, because of solubility limitation, precipitates as crystals in the organs, and this leads to toxicity. And hence the parent drug needs to have adequate solubility for the drug to be still bioavailable and also safe. And I'm going to quickly go through prodrugs to improve solubility. The first example I'm sharing you here is a phosphate ester prodrug of amprenavir. It's a fosamprenavir Lexiva, and this drug was approved in 2003. And the bioconversion happens through alkaline phosphatases that are present in the gut wall. And in the case of fosamprenavir, compared to amprenavir, it has a 10 times higher aqueous solubility. And this enabled simplified formulation that has a much better patient compliant regime. Amprenavir by itself was originally formulated in a capsule formulation using very complex milieu of excipients, including vitamin E tocopherol. And the patients had to take 8 capsules of 150 milligram. Whereas when formulated as an fosamprenavir, we were able to achieve much better profile, and patients had to take only 4 tablets a day, and it is much better delivery process. And in the case of the prodrug, because the prodrug was discovered later, it had the advantage of additional patent exclusivity of up to 6 years, and I believe the most recent patents on the fosamprenavir have expired now. And in the case of prednisolone phosphate, again a phosphate prodrug, it's having it as a phosphate prodrug enabled the liquid formulation, and we were able to dose it as a liquid formulation for children as well. So for the phosphate prodrugs to be activated, it is activated through alkaline phosphatases that is present in the small intestine, in the villi of the small intestine. And here I'm showing a zoomed image of the small portion of the intestinal wall, where the prodrug is then activated by the alkaline phosphatase and releasing the parent drug, and then that is then further taken up for the into systemic circulation. So as you can imagine, in the case of the phosphate prodrugs, if the prodrugs get cleaved prematurely, and you still have the parent drug in very high concentration, and that may still have the high permeability through the gut wall to be taken up for the systemic circulation. However, if the supersaturation is high, there is a risk of the prodrug, of the parent drug precipitation, in the prior to systemic circulation, making the phosphate prodrug not effective. Just wanted to kind of summarize that. In the case of oral etoposide, there was no clinical offering, advantage of the oral phosphate prodrug. And in some cases, phosphate drug of the Taxol was tried, very poor enzymatic conversion of the alkaline phosphatase. In the case of the Taxol, phosphate prodrug of the Taxol, the phosphate moiety is shielded sterically in the molecule, and hence it was not accessible for the alkaline phosphatase to activate the drug. And I already spoke about the premature cleavage and precipitation of the insoluble salts. In the case of the TFOs, it was dosed as a prodrug, as a phosphate, and then in the presence of the calcium salt that is taken as a proton pump in antacids, it can precipitate the drug can precipitate as a calcium salt. And so we have to be mindful of these challenges when we develop oral phosphate prodrugs. So recently, there has been a very exciting development in the case of delivering compounds with poor aqueous solubility. What I'm showing you here is a compound. It's a product that was approved in 2015. Isavuconazonium sulfate. Crizamba is a marketed drug. It is an antifungal drug to treat invasive aspergillosis and mucormycosis, and it's a novel sarcosin ester prodrug. So what is shown here is the sarcosin ester prodrug of the azonium moiety here. And in this case, in the esterases convert or cleave the amidate group here to give the primary alcohol. The primary alcohol then further undergoes intramolecular cyclization and resulting in the amide cleavage here, releasing the isavuconazole. And these are what are called self-immolating prodrugs. Essentially, you have the primary alcohol attacking this to form the cyclized moiety here, and then subsequently cleaving the amide bond here to give you the drug. So this has been a very interesting development, and similar strategies for improving solubility could be tried for similar compounds. And likewise, Selexipag is another new product that got approved in 2015. This is a prostacyclin receptor agonist to treat the pulmonary hypertension. And this is a novel acyl sulfonamide prodrug. And the acyl sulfonamide moiety makes prolonged release of the active moiety in circulation. And this essentially makes the drug safer because it modulates the peak to trough fluctuations of the parent drug because of the prolonged duration of action of the prodrug. So when do solubility enhancing prodrugs work or needed? When the parent drug has very reasonable permeability but poor solubility, essentially BCS class 2 or DCS class 2A, class 2B compounds are the ideal candidates for solubility enhancing prodrugs. And in cases where the compounds are not highly metabolized in the liver, again, as I spoke about in the case of the permeability enhancing prodrugs, if the first pass metabolism is significant, then the solubility enhancing prodrugs would still not be effective. And these are particularly important for compounds with very high dose number. We spoke about it earlier as well. And compounds with very high dose number can benefit from solubility enhancing prodrugs. So I want to just quickly talk about the carrier-mediated absorption. In this case, an example I'm sharing you here is the L-valyl ester of ganciclovir. In this case, the bioconversion happens to the pepti-1 transporter in the intestine, and this is transported from the pepti-1 transporter, and then it is cleaved in the liver in the hepatic esterases to release the ganciclovir. And the oral bioavailability enhanced from 6% for ganciclovir to 61%. And this is because of the carrier-mediated transport to the H-pepti-1 in the intestine. Likewise, there are other prodrugs for targeted delivery. The entire area of tumor targeting, ADEPT and GDEPT technologies strategies can be considered as targeted delivery of anti-tumor agents. And liver targeting, two examples that I'm sharing you here. One is the predofovir mesylate, and here it undergoes in the liver through CYP450 catalyst enzymes to release the phosphate predofovir. And simvastatin again is an example of where the inactive lactone converts to the hydroxylic acid and in the liver to release for it to be bioactive. So when we consider prodrugs, there are multiple challenges in formulation, that we have to be mindful of. One is the chemical stability. The drug pro-moiety linker stability is very important, and it has to be stable enough to be synthesized, but labile enough to be released as a parent drug in vivo. That is very important. And the potential for formation of the byproducts, and that is very important. I spoke about the pivalloyl ester, pivampicillin, and the pivalic acid that is generated has been shown in some cases to inhibit the carnitine. It's a protein that is intracellular. That inhibition of the carnitine results in cardiovascular events, and these types of pro-moiety safety needs to be well understood in the case of developing the prodrugs. And of course, the other aspects such as the solubility of the prodrug itself and the polymorphism of the prodrug are also important considerations that we have to pay attention to. And last but not the least, the formaldehyde, in some cases, we have found to interacts with the gelatin capsules. And so, that also it's one of the challenges that we have seen in developing some of the prodrugs that release the formaldehyde from the formulation. So in this case, I'm just sharing with you tenofovir disoproxil that I spoke about earlier. And tenofovir disoproxil undergoes hydrolysis, releasing the formaldehyde. And in the presence of formaldehyde and another molecule of the tenofovir disoproxil, it forms the dimer. And this is an impurity that is generated when the molecule is subjected to hydrolysis. So what this means is that we have to be mindful of controlling the hydrolysis, essentially protecting the product through maybe a film coating or a barrier coating to prevent the hydrolysis reaction from happening. Likewise, in the case of enalapril, we have a hydrolysis reaction leading to the enalaprilat, and this undergoes a cyclization and to give the diketopiperazine of the enalaprilat, and that is again another side reaction that we have to be mindful of when we are developing the prodrug of enalapril. So when we speak about prodrug development, there are multiple in vitro and in vivo studies that we use for prodrug characterization. So what I call it as a multi-parametric optimization challenge. It can be solubility, permeability, stability, and bioconversion, and also pharmaceutics and toxicity. These various assays, whether it is pH solubility, solubility in physiologically relevant media, or whether it is various permeability assays, either it is PAMPA, MDCK, cell lines, KCO2, and the cells that express the transporters, and also some cases using the portal vein cannulated animals to understand the permeability of the drugs, are also some of the assays that are used to understand the prodrug properties and whether it is functioning as intended. Stability of the prodrugs are typically evaluated through various assays, including intestinal S9 fraction, and evaluating through other hepatocytes and purified enzymes, and even in some cases where the tumor targeting is intended, using tumor cells as a way to screen for bioconversion of the pro-moieties are also important. And understanding the pharmaceutical properties, whether it is a crystal form, dissolution, particle size, toxicity profile of the drug, these are all the various aspects that need to be paid attention to while developing prodrugs, successful prodrugs. So I spoke about many of these in terms of the challenges associated with the discovery and development of prodrugs. Synthesis of the prodrug itself can be complex because of the complex chemistry. And also as I spoke about earlier, the compounds have to be stable enough to be synthesized, but labile enough to be converted in vivo. So that is a tricky balance that we have to play. And sometimes, as I spoke about in the case of Taxol, the pro-moiety, if it is sterically hindered, synthesis itself can be significantly challenging. And we spoke about the controlling the site and rate of bioconversion is very can be challenging. And quite often, in the case of prodrugs, we do not have appropriate animal models, preclinical models, to understand the efficacy of the prodrug because of the interspecies differences. This is particularly important in the case of drugs that are metabolized by specific enzymes, and how the rate and the expression of these enzymes, the preclinical species, rats and dogs, are very different from human, and hence understanding the prodrug performance in the preclinical model and scaling that to humans can be particularly challenging. And analytical scientists can appreciate this. Profiling of the prodrug, parent drug, and the metabolites thoroughly through the analytical methods can be particularly challenging. And unlike the single moiety, doing a pharmacokinetic modeling, physiologically based pharmacokinetic models are difficult to develop in the case of the prodrugs. And spoke about the safety assessment of the prodrugs. As an example, I spoke about the prevail oil ester prodrug, prevalic acid generation. And also the regulatory pathways needs to be fully understood. Particularly if you are working with the products that have been already later in development or already marketed drugs. So kind of want to summarize and want to kind of what I want to impress upon today is that in order to develop a successful prodrug into market, it is a multidisciplinary effort. And we need scientists with a very specific skill set, expertise in skill sets. We need specialist batsmen, specialist bowlers, wicket keeper, and ably managed by a captain. And so that is what the prodrug development effort is all about. And we need scientists with very strong synthetic chemistry skills, and a thorough understanding of biology, metabolism, and pharmacokinetics is very important. And biopharmaceutics understanding of the prodrugs is very important for us to develop good prodrug, and understanding of the safety and toxicology. And I would say a chemical modification of the drug is a creative process, and out of the box thinking of various possibilities is very important to come up with interesting prodrugs, as I have shared in some of the examples today. And last but not the least, prodrug efforts is all about persistence, perseverance, and patience to give prodrugs that addresses nations of the needs of the patients all over the world. So with that, I thank you for your attention, and I would be able to take some questions in the remaining time. I'm available for some more time as well.
Thank you, Dr. Sridhar, for your wonderful presentation. I believe the information you have shared is very useful to our participants. We have a couple of questions. One is from Dr. Ravi and one is from Dr. Amit. So I would request Dr. Ravi to go ahead.
Thanks, Javed. So Sridhar, very wonderful presentation. I have one question. In NIPER, we may be having drug discovery molecules, but at what stage we should be deciding that, okay, prodrug will require or not require? I can give more specifics to that question. We may be working on a CNS of, say, Parkinson or Alzheimer, where the molecule has to be given orally because of a patient compliance. But certain molecules are giving good activity in vitro, definitely on not in vivo, but definitely in vitro.
Mhm.
And the enzyme assays, the in vitro cell culture, certain biomarker assays, and other things is positive. At
Mhm.
what stage we should switch over to a prodrug? If you can put light on based on your experience.
Yeah, very important question, Ravi. I think when I speak about the prodrugs, I mean, think about the target organ, right? The target organ is the deciding factor as to how you want to prosecute your candidate molecule. In this case, you're speaking about a drug that is a target organ is the brain, and so to understand how delivery to the brain is effective, you need to have the appropriate preclinical model with the drug, and when dosed orally, are you able to see the drug uptake in the brain? And many drugs that are intended for the CNS targeting do not cross the CNS barrier, blood-brain barrier. And as you may be familiar with, there are transporters that have been leveraged. Example I can think of is the levodopa. Dopamine itself is not directly does not cross the blood-brain barrier easily. Whereas as the L levodopa through the L amino acid transporters, LAT transporters that is in the blood-brain barrier, makes the levodopa to be delivered to the brain. Droxidopa is another example. Again, similar LAT transporters that is used. And in a sense, understanding the delivery to the brain, and also to in a broader scheme of things, understanding the delivery target organ, delivery to the target organ would be a very important point for you to evaluate the need for the prodrug. And of course, we spoke about if it is solubility limited, then right away that you're going to have only certain leverage that you would have by the standard approaches for solubility enhancement. In those cases, if the standard amorphous formulations of delivery from solution is still not high enough, then you need to think about the prodrug modification to improve the solubility. And this is true both for the orally administered drug and also for in the case of the orally administered drug, permeability also becomes a challenge. And so to have a good assay on the permeability would give you an indication on whether a prodrug that enhances permeability is needed or not.
Thanks, Sridhar. Thank you very much, Dr. Sridhar, for the very brilliant talk. I have a few questions. My first question is, you spoke about sulfonamides as prodrugs. So I request if you can throw some light that after bioactivation, what sulfonamides are converted into?
It depends on the drug that we are working on, right? So the sulfonamide prodrug would be essentially, you would have the sulfonyl moiety would be the one that you would have becomes the active moiety.
Okay. And my other question to you is that while we talk about several prodrugs, so you also spoke about several drugs which the prodrugs which are being acted upon by esterases. So is there any disease-wise classification or some biasness towards a particular enzyme site? For example, if we are targeting tumors, so we must be having the drugs with let's say ester linkages, or let's say if we are making some antibacterial, we should have some sulfonamide type of moieties. It may be a very bizarre question, but I would like you to throw some light upon it.
Yeah, so tumor targeting is a whole big area. I didn't have an opportunity to talk about it today. Essentially, tumor targeting, if the tumor cells have specific enzymes that are overexpressed, potentially you can create a prodrug that are activated only by those those esterases or those enzymes. The ADEPT and the GDEP technology that I spoke briefly about today would essentially fall under that category. tumor targeting with specific enzymes that are overexpressed in the tumor cell lines is again an approach for delivering drugs to the tumors. It is fairly well understood. I think you may want to look up some references in that area. You'll see a lot of very good literature.
Thank you. Thank you very much.
Now I would like to request Dr. Amit for a formal vote of thanks. Thank you very much, Dr. Sridhar. Your talk on prodrugs in drug discovery has really enlightened us, and several questions that were being raised were wonderfully addressed. But one thing I would like to state, and I was also discussing it with Dr. Ravi, that with your expertise and very keen wisdom in the field, we miss you in person. So once you are there in India, so I request that you pay us a personal visit so that we can learn more and more from you in a more informal setting. So this is my request to you. And also I would like to thank you on behalf of NIPER family that you are always there at our disposal as and when required. And also you are waking up too early in the morning in US. So I think you deserve a round of applause for that also.
So thank you very much, Amit. I really appreciate you, saying very kind words, and I really look forward to visiting. I do visit Ahmedabad. Piramal has a development center, and when I visit Ahmedabad next time, I will make sure that I visit all of you. It's been a pleasure to interact and work with Ravi over the many years, and would be happy to meet the academic circle in person. Wonders of the modern technology makes these types of interactions possible. Pre-pandemic, maybe we would have not even thought about doing this through over Zoom. And so that is the beauty of the modern technology. And so, in a way, it is a blessing in disguise for us to be able to interact in this format.
Certainly. Thank you very much, Dr. Sridhar, once again. And now we close the session.
Yeah, thank you so much. Yeah,